Good morning student. Welcome to Sheba online class. Last two sessions we are going to monocot stem, monocot root, dicot stem, dicot root. Now we are going to monocot TS of dicot leaf. Okay, TS of dicot leaf is a transverse section of dicot leaf. So first upper and lower epidermis. Two epidermis are present. One is upper and another one is a lower epidermis. First we are learn the upper epidermis. Upper epidermis is the outermost of the cell, outer layer of the cell. That is a single cell formed. The parenchymatous cells are formed, arranged. Next, this is made up of only for the parenchyma cell. This is called upper epidermis. The cuticle layers are present the upper side. Okay, first one is a epidermis. Epidermis is a outermost layer. This is a single cell form. Parenchyma cells are present in the upper epidermis. Then outermost layer cuticle will be present. It's clear? Next, lower epidermis. Lower epidermis are single cells. Lower epidermis are single cells and parenchymatous cells. The cuticle will be present of the lower epidermis. So first upper epidermis, the less than stomata are present. Less than stomata are present upper epidermis. Lower epidermis, the number of stomata present. Lower epidermis. It's clear? Are you going to understand? Upper epidermis, less than stomata are present. Lower epidermis, more than stomata are present. Okay. Next, this is a two cells are differentiated to the mesophils. What is the mesophil? Meso means is a middle. Mesophil. Meso means middle. Okay, center. Phil means leaves. So middle leaves are arranged the two leaves. One is two sponges. One is a palisade parenchyma and another one is a sponge parenchyma. Understand? One is a palisade parenchyma and another one is a sponge parenchyma. So this palisade parenchyma is a elongated shape. Elongated. Palisade parenchyma is a elongated shapes. At present there is a chlorophyll. Chlorophyll will be present the center of the point. Chlorophyll present the center of the point. Okay. Then lower side, the same time lower side sponge parenchyma will be present. Sponge parenchyma will be present. This is a intracellular spaces, intercellular spaces parenchyma. Then stomata, less in the stomata. What is the function of stomata? This is a gaseous exchange form. Gaseous exchanges form of the stomata function. So what I tell? Mesophil, upper and lower epidermis is called mesophil. There are two different types of cells formed. One is a palisade parenchyma and another one is a sponge parenchyma. It's clear? Next, the center of the portion is a vascular bundle. You know last class vascular bundle? Xylem present, phloem are present. This is a metaxylem and protoxylems. What is the function? They are conducted to the water and mineral salt. Then phloem is a storage of the food. It's clear? So centermost of the point vascular bundle is presented. Upper side xylem present and lower side phloem are present. It's clear? Okay. Once again, upper two epidermis are present. One is the upper epidermis, another one is a lower epidermis. This is a single cell, outermost of the cell. This is a lower epidermis is a single cell formed. Stomata are formed and then lower cell of the oval shaped. Listen, upper side cells elongated. lower side or oval shape. Here, two differentiated from the cells. It's clear? Are you going to understand? Next one is here. Palisade parenchyma, all are only arranged in the parenchymatose of the cell. Parenchyma, chymatose of the cell. The stomata, this is a less in number, only the upper side. So lower side of the many are present in the lower side. Next, this vascular bundle is here. Last previous class your vascular bundle are present there. All the monocot and dicot, monocot stem and dicot monocot root, all are vascular bundle. But the leaves is present only. What are your bundle sheath? Or telling the bundle sheath only. Mention the vascular bundle. At inner side, the present of the protoxylem, metaxylem. Okay. Next phloem is here. What the phloem? This is here. This point is here phloem. This one is here xylem. So the cuticle or outermost layer, the cuticle will be present. So what is the cuticle? The cells are cuticalized, and then is here waxy substances. Jelly-like substance is doing the cuticle layer. Okay. Next, these are all the tears of dicot leaf. Once again, I tell the oral. Upper epidermis, two outermost layer of the upper epidermis and lower epidermis. This is made up of parenchyma cells. This is a single layer, and then all are closely arranged. The single layer parenchyma cells, the upper side is here cuticalized or formed. So contain the numerous of the stomata in the lower side. The stomata is here alpine, gaseous, less than water vapor. Okay. Next is a palisade parenchyma. Mesophils are differentiated two types. One is a palisade parenchyma and another one is a sponge parenchyma. Palisade parenchyma is elongated and chlorophyll are present. What purpose of the chlorophyll? This is a photosynthesis. Is doing the photosynthesis. So that is called the palisade parenchyma. Okay, it's clear. Now that same pattern. Next, tears of monocot leaf. Okay. First, the tears of dicot leaf. The same method of the tears of monocot leaf. This is the upper and the lower epidermis will be presented, but one is a variation. This is single cells are formed, but the all cells are is a one or two cells or large cells will be present in the monocot leaf. So that is called the pulliform cells. Pulliform cells are present only the monocot stem. Look here. So this is a pulliform cells absent of the tears of dicot leaf. Monocot leaf, pulliform cells are present. Single layer of cell, one or two large cells. So that is called pulliform cells. Next, outermost of the layer, the same procedure. Cuticle will be present. Cuticles are a waxy substances, is a jelly-like structures. Okay. Next, upper epidermis. Listen, the upper and the lower epidermis is different. It is not differentiated palisade parenchyma and sponge parenchyma. It is not differentiated palisade parenchyma and sponge parenchyma. Next, upper epidermis, the chlorophyll are present in the cells. Lower also present in the cells. Okay. Next, phloem. Phloem will be most of the 1 or 2, 3 vascular bundle sheath are present. 2 or 3 vascular bundles are present. So it is a single vascular bundle. Vascular bundles only present. Upper side xylem, lower side phloem are present. So cuticles are present in the lower sides. This only for the palisade and parenchyma and sponge parenchyma. This is differentiated for the not differentiated for the cells. So this is a different, this is a elongated and the oval shape, but this is a not differentiated from the palisade and sponge parenchyma. All are arranged the same only. Okay, next vascular bundle. Vascular bundles are the conjoint and collateral is a xylem and phloem all are present in the bundle sheet, inside the bundle sheet. Conjoint and collateral, xylem, phloem all are present in the bundle sheet of the vascular bundle. Okay, it's clear. So differentiated from the dicot and dicot leaf and monocot leaf. So upper side, this is only for the upper epidermis, lower epidermis are present. Upper and lower epidermis are present. First one. Second is a palisade parenchyma elongated. Sponge parenchyma or palisade parenchyma is a differentiated shape. Is a monocot leaf, not differentiated palisade and sponge parenchyma. Okay, chlorophyll present and then chlorophylls are present. Pulliform cells absent. Pulliform cell present. Okay, monocot leaf, pulliform cell present. Pulliform cells absent. Next one is here number of stomata. Number of stomata, this is a less than stomata. Cuticle are present. Cuticle are present. Okay, xylem, phloem are present and then same xylem, phloem are present. Next one is a not very important one, not differentiated, non-differentiated palisade and sponge. So this is a differentiate from the palisade and sponge parenchyma. It's clear, palisade and sponge parenchyma is clear. Okay, first of all 5 mark question, very important question for the public. Tiers of dicot leaf, draw and explain that all. Listen student, you are every day well practice the drawing. Again and again you draw the diagram and label the parts. Very important portion of the dicot leaf and tiers of monocot leaf. Next we are going to plastid. What is the plastid? So that is called photosynthesis region. Plastid is a photosynthesis region. This is a oval in shape. The chromoplast of the chromoplast, lipid, thylakoid, stoma, granula, starch, these are all present in the plastid. This is only for the photosynthesis region. Green pigment only prepare the plastid. So this is the coil one by one is arranged. So that is called stoma. That is called. So single layer is a thylakoid. So that is single form is a thylakoid. So these are all stoma. Okay. Lipids are present. Is a fully content of the green pigment, chlorophyll. So that is called photosynthesis region, plastid. Next is a fully like the jelly-like structure formed. Fully jelly-like structures in between the jelly-like structures. Next one, important point of the chloroplast. 70 contain the DNA and is a ribosomes are formed. Is a stoma matrix. Matrix means a jelly-like structure. So that is called a matrix. Matrix present inside. Matrix present inside. These are all matrix. Okay. Next, this is a only for the chloroform contain the DNA 70S ribosome. DNA and 70S ribosomes. Ribosomes are present in the center of the point. Next, lipids. Lipids are present. And then starch granule. Starch granules are present in the inner side. So that is called plastid. Okay. Next, what is the photosynthesis? Photosynthesis is a prepare the sunlight and the one food for the sunlight. For example, photosynthesis equal to 6CO2 plus 12H2O gives light plus chlorophyll. Light plus chlorophyll will be light supported, light and chlorophyll. Okay, then gives C6H12O6 plus 6O2. So that is called the photosynthesis. What is this? This is a 6CO2 plus 12H2O light that gives the chlorophyll C6H12O6 plus 6O2. So first one is a carbon dioxide. Carbon dioxide. Then next one is a water and light chlorophyll C6H12. What is this? Glucose and oxygen will be released. So that is called photosynthesis. Next we are going to the mitochondria. Next one is a mitochondria. Mitochondria is the powerhouses of the cell. Very, very important. Listen, you are studying 6th to 9th standard lower class, you are studying the mitochondria. Okay, what is the function? The powerhouse of the cell. Mitochondria is the powerhouse of the cell. Ribosomes are, this is a crystal-like structure. This is a disc-like structure will be formed the mitochondria. So centromost matrix is filled with the matrix. Is a jelly-like structure, the same. Okay, ribosomes are formed. These are all the chain link structure. Ribosomes are formed only for the mitochondria. Next, inner and outer and inner membranes, double membranes will be formed. Inner and outer membranes are formed. So crystal nature. What is the crystal? It is a disc-like structure, finger-like structure. So that is called crystal nature. Okay, it's clear. So first main thing is a powerhouse of the cells of the mitochondria. Then axisome. What is the axisome? Axisome is the single crystal form of the axisomes. So the axisomes, the inner mitochondria. Listen one time. Inner mitochondria is a axisomes. Axisomes is a inner mitochondria is a one part. Next, the membrane where the regularly speed the tennis racket shaped. What shape? Tennis racket shaped of the axisomes. So these are all only the ATP synthesis. These are only the ATP of the synthesis. What is the ATP? Adenosine triphosphate. ADP, listen, ADP and NADP, ATP, then ADP and then NAP. Okay, these are all the NAD. Next, NAD. ATP means adenine triphosphate. ATP. ADP, adenosine triphosphate, adenosine diphosphate, ATP, adenosine triphosphate, ADP, adenosine diphosphate. Next, NAD, nicotinoid, adenine dinucleotide. Okay, NADP, nicotinoid and adenine diphosphate. These are all the reaction of the photosynthesis. Two react. One is a light dependent the photosynthesis and another one is a light independent of the photosynthesis. Okay, it's clear. Next, the Melvin Galvin is this is a American biochemist. Melvin Galvin is a American biochemist. chemists. So he is discovered the pathway of the photosynthesis. Who discovered? The Melvin Galvin, the American biochemist, is discovered the photosynthesis. It's clear. Next, the cycle is from the Galvin cycle. You are going to further class study 11th 12th, or you explain the C3 carbon, C4 carbon, then you one by one you are study the detailed. Okay. So that is here Galvin cycle is here only discovered the Melvin Galvin. Okay. Next, this is here artificial photosynthesis is producing the renewable to energy. Artificial photosynthesis. Who discovered? This is here sunlight Indian of the Rudas Bharat Rans is discovered the artificial photosynthesis. Okay. Next is a respiratory quotient. Respiratory quotient of the ratio will be volume of the CO2 liberated and volume O2 consumed. RQ, listen. RQ, respiratory quotient. Respiratory RQ equal to volume O2 liberated, volume CO2, volume of CO2 liberated, volume of CO2 liberated and volume of O2 consumed. So that is called respiratory quotient. Respiratory quotient is the ratio of the volume carbon dioxide liberated and volume of the oxygen consumed and during the respiration. So it is called expressed as a RQ. RQ means respiration quotient equal to volume of the CO2 liberated and volume of O2 consumed. Oh, it's okay, it's clear. Next, anaerobic respiration. What is anaerobic respiration? The anaerobic respiration is takes place with the oxygen. Without oxygen, glucose is converted into the plant. Okay, C6H12O6 plus 2CO2 and then gives 2C2H2OH of the energy. That means ATP, adenosine triphosphate will be came to out. So that is called energy. Listen student, now third session is over. The first lesson you are all sent the homework is properly, and the first lesson is very important lesson in the public exam. Every test and quarterly, halfly, annual, every test will become to the first lesson. Is a theory and practical is very important lesson. All are read well known. Thank you student.